In this lesson, we'll take a look at functional magnetic resonance imaging. This is a widely used technique in the neurosciences to measure the relative brain activity while people are doing different kinds of tasks. It produces an image like this, where you'll see this is a person looking this direction, and their skull is removed. Here we see the surface of the brain, the cerebral cortex, and a cutaway view of that. So the fMRI will give us a picture of the structure of the brain, but in addition, superimposed on that, these color splotches indicate relative levels of activity of certain regions of the brain. So red might be assigned, these areas in red show high activity relative to other areas of the brain for the particular task that the person was doing. Yellow might be a little less activity, green even a little less. Because the activity is shown here in the back of the brain, we know this is visual cortex. This person is likely doing a visual task. So the fMRI technique is showing not only a structural view of the brain, but also a functional view of the brain. Now, how does it measure the functional aspect of brains? To understand that, we have to know a little bit about brains and their blood supply. What you're looking at here is an exposed portion of the cerebral cortex. This is the right side. The skull has been removed, and the kind of whitish-grayish areas here, this is the surface of the brain. Now, these big tubes here are the blood vessels, right? So the brain is very metabolically active. It uses a lot of glucose and a lot of oxygen to support the electrical activity that it's constantly generating. The key thing that the fMRI technique takes advantage of is that when a particular part of the brain becomes activated, it recruits an increased blood flow to that localized region. So let's pretend that this person is not undergoing surgery, but instead they're in the fMRI machine, right? And let's say whatever task they're doing, it is activating region with number 3 here. But it doesn't activate region number 10. Now the idea is, because the brain cells in region 3 are going to show elevated electrical activity, they're going to signal the nearby blood vessels to increase the blood flow to this region. And the fMRI machine can detect the increased blood flow to this region. Because area 10 was not involved in the task, you don't get a signal change here. You don't get extra blood flow here. So the key to the technique is measuring changes in blood flow, which means that fMRI is not directly measuring brain activity, it's an indirect measure. Changes in blood flow are a proxy for brain activity in a certain region. Now here we see another picture illustrating this effect. Here we have some brain cells here called neurons and the blood supply. Now the red dots would be oxygenated blood, so these red blood cells have hemoglobin that has oxygen attached, and the blue dots here would be the red blood cells. They've already given up their oxygen, so oxygen is going to diffuse out of the blood into the brain cells. Glucose also will get into the brain cells. So when these cells are just kind of at rest, they use some amount of oxygen and glucose. But if that particular set of brain cells becomes active because of some task, they're involved in doing some kind of computational process, then they signal the nearby blood supply to increase blood flow, bringing more oxygen and glucose, right? And the fMRI machine, it can detect this change because the magnetic properties of oxyhemoglobin are different from deoxyhemoglobin. So because oxygenated hemoglobin and deoxygenated hemoglobin have different magnetic properties, the machine can detect changes in blood flow that are correlated to different levels of activity of localized brain regions. Now, because fMRI is measuring changes in blood flow, it's actually the signal that's recorded by the machine is a delayed signal. So up here we have the neuron, and if we could measure its electrical activity during some task, right, these little bars here would indicate it's making a lot of electrical signals. So the person is maybe doing some kind of task, this brain cell becomes highly electrically active, but the time course of the change in blood flow is delayed, right? So the fMRI technique unfortunately does have this feature built into it. It's a delayed signal because it's measuring the increase in blood supply to the neurons that show high activity, but the electrical activity is happening before the change in blood supply. So while this delay is not ideal, you can kind of just, in published reports, what the researchers do is they just sort of correct for the delay. So you can, kind of estimate and actually measure what is the delay, and then sort of just take account of that when you are showing your results. And the fMRI signal is called the BOLD signal, standing for blood oxygen level dependent. Okay, so let's return to the example of the subject is in the fMRI machine and they're doing some kind of visual task. And I've shown you here an example of the BOLD signal here showing elevated activity in the visual areas of the brain for the visual task. But how is it that you only find activity in the visual part of the brain? Isn't the subject also processing sound and they're feeling themselves laying in the machine? After all, the fMRI machine is a very loud machine, so there's all kinds of other things going on in the brain. The person's having thoughts, maybe they're feeling their body, they're hearing noises. How is it then that you can show activity just specifically for the visual task when there should be all kinds of other things going on in the brain? And the way they do that is by using a subtraction technique. And here's how this works. Okay, so let's take a look at this image here. Now this subtraction technique is used for fMRI, but also for the earlier technique called the positron emission tomography, but we're not going to get into that technique. So imagine this is an fMRI study. So here's a view of the brain, and again the coloration would indicate widespread activity in the brain, right, on the cortex of the brain. And again, let's imagine we're having the subject do some kind of visual task. So they're in the machine, they're looking at a screen, and different images are coming up on the screen, so they're processing, they're viewing these visual images. But of course, while they're viewing the visual images, they're also hearing the machine, they're maybe having other thoughts, or they're remembering something, and they're feeling their body, etc. So that's why you get all kinds of activity in the brain. But what we want to determine is what parts of the brain are specifically active for a visual task. So what you do then is during another measuring period, you simply measure the person's brain while they're doing a control activity, a control task. In this case, it might be just laying there in the machine. They're still hearing things and doing all kinds of other stimuli going on, but what they're not doing is the visual task. So they might be just looking at a dot on a screen or just looking at a blank screen, right? So yeah, there's going to be widespread activity in the brain. But what you can do then is take this activity in the control period of measurement and subtract it from the activity measured during the task, in this case a visual task, and the result of that subtraction are just those parts of the brain that are active for that specific visual task. And you might do this with several subjects, and then you average those subjects to get a sort of final averaged difference. Okay, so now let's apply what we know about fMRI as a general technique to Kanwisher's studies of face perception. So when she presents a graphic like this, in her question, does the human brain contain a region specialized for face perception? This little greater than sign here is an indication of the subtraction method, right? So she's going to be measuring brain activity while subjects are looking at faces, and then subtract the brain activity when subjects are looking at objects. So there's going to be lots of areas of the brain that are going to be active for both processing faces and objects, and the idea is we're not interested in those areas of the brain that are jointly activated by faces and objects. We want the areas of the brain that seem to be specifically activated for faces. So you subtract the activity for objects. So they call that a contrast, but theoretically it's this subtraction technique. Now what the machine does, the fMRI machine does, it's taking pictures of slices of the brain over time. And now if we focus on this little red dot here, this would be one little region of brain called a voxel, a small little volume of brain over time. And so the machine is simply measuring the relative blood flow, remember what, right, which is a proxy for electrical activity of brain cells in that region. So that it's just going to be measuring a signal that is related to brain activity for that little bit of brain. Of course, it could be doing it for all these other bits of brain too. But one little volume is called a voxel. And if you look at the signal from one little voxel over time, it's got a lot of noise in it, right? And so that doesn't look like anything that is immediately able to be interpretable, right? But what you can do is you can do some statistics, and there's lots of statistics involved in analyzing this fMRI data. So the basic statistical question is the following: is the signal higher for faces than for objects in any particular voxel, right? So here we have the person in the machine is seeing an object on the screen, and that little bit of brain tissue, that voxel, had this signal recorded. Then the face stimulus comes on, and this signal is recorded, right? And the object comes on, this signal is recorded, and then a face and this signal. And so what the statistical technique does is just ask this basic question. If you kind of just average the activity for faces and objects, will you find a difference in activity in that particular voxel? So when Canwisher shows us some data like this, she has found that this area here in the fusiform, the right fusiform gyrus, and we're going to call it the fusiform face area, right, shows significantly increased activity for faces over objects. Faces over objects. Now notice there is a little bit of activity more than baseline, so this would be looking at a blank screen, for example. So objects do turn on the face area a little bit, but she's going to argue that the significant difference here indicates that this area of the brain is selectively processing faces. No other type of stimulus will activate this area of the brain as much as faces. So just to recap, this signal here for the face here might be an average of several periods from that one voxel within the face area. But when you average together lots of presentations of faces and objects, you get a nice display like this showing clear selectivity of that region for faces versus objects. So at this point you can pause the video here and try some retrieval practice questions.